Thank you again for the invitation, for the words, kind words of years ago to you. It is a great pleasure for me to have this opportunity. I think the microphone I need. Without microphone, serendipity is an interesting term. I think one of the reasons that it is worthwhile to go in the direction to be a researcher is this word, serendipity. I have given this lecture, which you will hear here at my company some weeks ago. We had a journal club, and in this journal club we give some lectures to each other and analyze some journal articles. And I've given a different word for this. If I wanted to translate it into Hungarian, and if I translate it from the Hungarian version, I would give unexpected term, but it is not really true. And it's a complicated term, nevertheless, I think it is interesting to speak about the term and the origin of it. I found in the internet the following definition, which you can read here: the occurrence and development of events by chance in a happy and beneficial way. I think this long definition is valid, but at the same time, you can understand why to use serendipity, because it's a simple word, and this simple word describes this rather complicated phrase. And chance discovery they also use sometimes, but it is something more. Serendipity has a capacity, preconditioning is a prepared mind, who is able to recognize an event which is positive in this activity. The etymology is also complicated. It is a Persian word, the name of Sri Lanka from previous Ceylon is Serendip, and there are tales about the three princes of Serendip, and it was taken in use when the English writer who used this term the first time. In my lecture, I wanted to a little bit analyze the so-called target-based drug discoveries, which is characteristic for the drug research the last decades. And I selected some stories, and it is the second part, where, interestingly, during the research work, even the targets changed. The first part, I'll give you an overview on the success to target discoveries, but there are only examples of this 50 years, so it can't be comprehensive. Here you can see this very short overview, beginning from spironolactone in 1994, and recently introduced very successful hepatitis C drug, If we start a relatively complicated steroid structure. Aldosterone is hormone. The structure was elucidated in 1954, and I think it was rather quick that could introduce 4 years later spironolactone as an aldosterone antagonist. It was a very important drug for cardiovascular diseases, hypertension, for the treatment of hypertension as diabetes. However, spironolactone has several side effects, hormonal side effects, and to optimize it with a derivative, with an analog, with plastic, some techniques. It was introduced by Pfizer, Then, in the 60s, early 60s, the beta blockers were, I think, the great development. Propranolol, the pioneer drug, in 1962, and then several similar structures which were non-selective. And the problem of the selectivity, that there are two subtypes of the beta blockers, beta 1 and beta 2, and the non-selective agents acts on the polycyclic receptors too, and it is a side effect in patient asthma or illnesses, and therefore it was achievement that to develop beta-1 selective agents as acetylcholes and if you look at the structures, there are very interesting characteristic structures, arioxy, propanol, amines. Even the amine substituent is identical or rather similar isopropyl or phosphoryl groups. Here, the beta selective blockers structurally are rather similar. They are nearly, one can say, substituent analogs of the previous ones. In the para position, you can see, or excuse me, point there, you can see there's a para position of the phenyl ring, phenol, using substituents can result in beta-1 selective agents. Ethanol was the most successful at the beginning. It doesn't penetrate the brain barrier, and therefore it is lack of CNS side effects. Metoprolol, I mentioned about from the research of Hessler. Hessler was a small company in Sweden, and long period, two decades, they hadn't enough other drugs as lidocaine in their portfolio. And one of the first discoveries was metoprolol, which came on the market, into the market as a beta-1 selective agent. Can you hear me back, or should I keep different the microphone? It's possible. As I mentioned before, these beta-1 and beta-2 subtypes can be used for research, pharmaceutical research, and starting from the structure of epinephrine in the English version, or adrenaline, were developed the salbutamol, beta-2 agonists, and the long-acting salmeterol and formoterol, which are very, very important drugs for the treatment of asthma and chronic obstructive pulmonary disease, which is COPD. If you look at the structures, adrenaline structure is interesting, has similarities. And if you compare salbutamol, a short-acting agent, with salmeterol, the lipophilic side chain is characteristic in the long-acting agents. Then we come to the 70s, a breakthrough discovery for the treatment of gastric ulcer disease, the cimetidine, from the research of Swiss Klein and French. And we are all grateful to Professor Robin Gannerin, who published a lot about the history and development of cimetidine, which was the first blockbuster, as we call this in the little slang, very successful drugs. Maybe I go back to the first photos. It was a small group 10 years ago, and you can see here the first row in the front, Professor Gannerin, and we are missing him here in the second short course here in Rio. And Paul Erhard, in the second row, you can see here the Pilgrimage of Proudfoot, myself, Bukon Chorgal, Professor Barillero, Antonio Monch, and Philippe Royer. Yeah, and also the students, I have to show you. The students on the short course. Okay, then we go back to the cimetidine. It is rather interesting, and it is characteristic for our life present, that we have a quick, sometimes I have the feeling, too quick life. It was the cimetidine story, it lasted 5 years, that they haven't found any real active agents. It's this kind of French. And it is my question, which company would allow or permit nowadays such a long process without any big achievements? And then came cimetidine, which was very successful, but before coming to the market, there were 2 lead compounds which were used on the clinic with failures. And you know, if I add these 3 informations, 5 years without results, and then 2 lead compounds in the clinic with failures, I think it's a real achievement that cimetidine became a success. And it was a pioneer discovery, which was followed by analogs, which optimized the H2 blocking activity, the ranitidine and famotidine, which cover nearly 2 decades research if you look at the years. The structurally they have similarities. Cimetidine has a cyanogranidine group, comparing ranitidine to lipoethylene diamine, and famotidine as sulfamoyl propyl imide structure. And all the compounds have a thiomethylimidazole or thiomethylmethyl and so on structure. Then at the second half of the 70s, coming the ACE inhibitors for the treatment of hypertension, the pioneer drug captopril, which is one of the nicest story of the drug discovery, coming from the natural product monopeptide, which was then optimized, and the first oral reactive agent was captopril, which was a great success in this field. It is very interesting that at the beginning such an agent could be used only very carefully and cautious conditions. It was not allowed only after all the other agents were used to make a treatment with captopril. Nowadays, they are basically the first agents which are used in the treatment. But it takes always many times. In this respect, it's a complicated issue to avoid any adverse effects which are not cannot be discovered in models. Then, nearly parallelly, were angiotensin II antagonists, the pioneer losartan and its analogs. If you look at the structures, they have similarity, but also great differences. The captopril, which was a mercaptor acetylcholine, is a little bit different from the others. In the medical use, captopril preserved the utility in emergency for short, because it's a short-acting agent. Enalapril, lisinopril, perindopril are long-acting analogs, and perindopril has the longest half-life. They are used for the chronic treatment of sometimes lifelong treatments of hypertension. Here you can see the structures of the angiotensin II antagonists. If you see the biphenyl tetrazole, or instead of tetrazole, the carboxylic moiety, it is characteristic of these very important agents. Then we are coming already to the 90s. Trastuzumab was a breakthrough monoclonal antibody for the treatment of HER2 positive breast cancers. And a new agent, a new derivative of it, is a drug conjugate, trastuzumab emtansine, discovered by Genentech, where targeted drug delivery is solved by a drug conjugate of trastuzumab with the help of a linker. Here you see the linker, and this is a Y-shaped symbol of the monoclonal antibody is then conjugated with the cytotoxic agents, which is mitomycin in this case. And we will see how it can help for the treatment of such cases where trastuzumab becomes resistant. At the beginning of the 90s came the discovery of imatinib, a breakthrough discovery for the treatment of cancer diseases. It is quite natural, rare disease, the so-called chronic myeloid leukemia is amounts to about 10% of the leukemias. But if somebody is diagnosed with this illness, it can gain a normal health-like life, and therefore it was a breakthrough discovery for the cancer disease, which was before this period so hopeless. And nowadays we experience a lot of new achievements in this field. And in case of resistance, an analog has been developed, nilotinib, which has high structural similarity. If you look at the structure of imatinib and nilotinib, which are both pyridopyrimidine and aminobenzyme structures. we call this analog structure and pharmacological fundamentals. In the 90s and until now, we see a great development in the drug research of diabetes. Exenatide, I don't know how the pronunciation, is a natural product which was introduced as a pioneer drug for the treatment of diabetes, type 2 diabetes. Is a GLP-1 agonist drug. It is used in a subcutaneous injection. Shortly afterwards, the Danish country Novo Nordisk discovered liraglutide, which is used in once a day treatment, and nowadays it is under development even longer acting derivatives in the phase 3. To show you how they look, it is a very simple modification, seemingly simple, is a modification of the two places of the amino acid chain, lysine and arginine, in the case to get the liraglutide, and acylating with the barbituric acid. Here you can better see the natural product, the native human GLP-1, and liraglutide, which is only two amino acid differences and the barbituric acid acylation. I wanted to demonstrate you that these complicated compounds could be changed with a relatively simple way to get really effective and useful drugs. Then some new achievements, for example, the recently introduced not tofacitinib. I mentioned Janus kinase 3 inhibitor by Pfizer for the treatment of rheumatoid arthritis, pericarditis, and it is a chronology, excuse me, it is a little chaotic, but it is a chronology of the last 5 decades. Came the orally active diabetes agents, the DPP-4 inhibitors, because it is a great advantage compared to the subcutaneous treatment that they can be used orally. The pioneer drug came from the H3 screening at Merck, and parallel, the first discovery came at the Novartis with the gluten, but they were a little bit late in this competition. It is also underway in phase 3, once weekly analog of this, the correlation between this DPP-4 and the GLP-1 is easy to understand. The N-terminal histodylalanine, the dipeptide, is cleaved by DPP-4, and its inhibition gives the same result as the GLP-1 agonist, because the GLP-1 has then a longer activity. Here the structure of vildagliptin, sitagliptin, and omagliptin, which are nowadays very successful drugs of diabetes type 2. Then relatively new achievement, nearly parallel research, starting from the natural product fluorescein, developed company Bristol Myers Squibb and Johnson Johnson and others, SGLT2 inhibitors. It is the abbreviation of sodium glucose transporter, and it is a great achievement. The fluorescein, the natural product, the lead compound, is non-selective. It has side effects and low bioavailability, and the analogs solve this problem. We cannot speak in these cases about fast followers because they are practically parallel research achievements. Here are the structures of these compounds. Then phosphodiesterase 4 inhibitor for the treatment of psoriatic arthritis is a relatively new drug. It was approved last year. Now that was the first part of my overview, and now I would like a little bit deeper enter such examples were really interesting how these drugs were discovered. 5 cases I will enter a little bit more detail. Drospirenone, a contraceptive agent, escitalopram, an antidepressant, ezetimibe, cholesterol lowering agent, lamotrigine, antiepileptic, and omeprazole, which is a proton inhibitor. Even nowadays, however, there are generic products, they have a big sales value, which shows how successful these drugs were. Let's start with the drospirenone. Schering in Berlin, researcher, wanted to make a new aldosterone antagonist starting from spironolactone. And their idea was that they introduced cyclopropyl ring in the ring B of the steroid and ring B. And this spironolactone was already under development. And it has 5 times higher anti-aldosterone activity as compared with spironolactone. And the hormonal side effects were lower. Their goal was achieved, seemingly. And then came to the human experiment, the human investigation, and they noticed the hormonal effect, which was unexpected, spironolactone, namely low doses resulted in reduction of testosterone level in men. And they discovered that this spironolactone in human, with the help of the delta 1 hydrolase enzyme, is forming drospirenone. Practically is saturated one double bond in the ring 1. And this drospirenone, after longer investigation, they found that it is an excellent progestin, which has some diuretic activity. And it was developed to give one of the most successful contraceptive agents. I think it is a very interesting example. It is described in the analogous drug discovery book, the first one. Then let's have a look at the very interesting Lundberg story, how they discovered escitalopram. It started with a chemical serendipity. The serendipity can be everything, not only biology, but also chemistry. They wanted to make a melitrazine derivative to improve the biological activity, and the ring closure reaction with sulfuric acid didn't give the expected ring which they wanted to have, but phthalate derivative was forming. It was not planned. And if they compare these agents, the melitrazine was a norepinephrine reuptake inhibitor, an unselective one. The talopram, which was formed by chance, was a selective and more potent, even more potent norepinephrine reuptake inhibitor. However, they had to start the clinical investigation because of its psychomotor side effects. Then they didn't know at this moment the Nuremberg people what to do. And for their help was coming a discussion with the Nobel laureate Arvid Carlson, who initiated at the company to switch to the serotonin reuptake, selective serotonin reuptake inhibitors. And it is again an interesting point. They used talopram as a lead compound. Talopram, which was a norepinephrine reuptake inhibitor, and nevertheless they used it. It was an explanation for it by the inventor in the book, that in-house collections, there were several analogs of talopram, which had already some SSRI activity. Talopram, if you see the structure here, talopram, optimization with substituents ended up to give citalopram. It is a characteristic cyano substituent, a quasi-para position in the benzofurane ring. It's also interesting that the two enantiomers of the S-citalopram have not the same activity profile. The S-citalopram has a S1 inhibition, and beside this, it has allosteric binding inhibition, binding on S2. And the isomer counteracts these activity, and therefore it was very useful to develop the S-citalopram for the next product after citalopram. What I wanted to mention in the serendipity case, that if they carry out the research, they wouldn't have selected citalopram, because there was an even more active agent among the compounds. And here you can see the structure. But it was not developed by citalopram, and it was afterwards very successful, because this agent, this analog, has no allosteric size binding. Therefore, it was very fortunate how they selected the citalopram. Then the third story is the azathioprine, coming from the Sherry Law, nowadays Merck research, as inhibitors of the cholesterol, as they planned to inhibit, they started the research with inhibitors of ACAT enzyme, which is an abbreviation of acyl coenzyme A, cholesterol acyl transferase. And there was some lead molecules from Sandu and Perk Davies research, which they had, and the first step was to try a ring closure derivative of it. And these other Tidinon derivatives, which you see here, showed a poor correlation between the ECAT inhibitor activity and the cholesterol absorption. And therefore the researchers switched to in vivo experiments and started the investigation of ACAT enzyme inhibition. And the in vivo experiments, they were using these in vivo results and received a clinical candidate, this sharing compound. And on the clinic, they investigated that the side chain hydroxylation is taking place in the molecule, and it was also used to further optimization. And you can see the product, the ezetimibe, here on the right side. The substituent was also replaced by fluoromethoxy. It has a standalone character, the ezetimibe. There is no class as such, but it is very important to inhibit the external cholesterol from, and it's a very important drug among the products. Now I go and continue with the lamotrigine. It is also an interesting story. It was supposed that the folic acid inhibition gives anti-epileptic agents, and there were some known compounds, phenobarbital, phenytoin, carbazepine, which we have had these correlations. The research, Boros Welcome research, used anti-malaria drug pyrimethamine as a lead compound, and interestingly, if they optimize, the anti-epileptic properties were not correlated with the antifolate activity. More active in the anti-epileptic in vivo tests, not necessarily were stronger antifolate active agents. And the end product of this optimization was lamotrigine. And lamotrigine, which is a successful anti-epileptic drug, has a different mechanism of action. Is a blocker of voltage-sensitive sodium ion channels, and nearly without any antifolate activity. Last but not least, we arrived at the story of the omeprazole. We will hear a lecture from your on omeprazole. I don't want to enter too detail this story, but it is very interesting. It was obvious at the beginning of the research that the gastric acid has a correlation with ulcer diseases. And it was a hypothesis that gastrin was responsible for this, and to synthesize anti-gastrin agents was, for example, the goal of the ICI researchers. And they synthesized thousands of compounds, but they couldn't be developed. At this stage, there was a known compound from the research of Servier, the spiritual thioacetamide, and the third compound. And the Hassel people, who were already successful with metoprolol, as I mentioned, and also the calcium antagonist, wanted to create a new pioneer drug discovery in this field. And the first idea was to get rid of the toxic side effects of the thioamide with the help of ring closure analogs. But it has some Hungarian aspect in my native country, because the first very interesting compound was here to see in benzimidazole thiomethylpyridine derivative, which was, however, in a Hungarian patent application. And the world leader of Hessler compound from Sweden came to Hungary, and then they could realize that the Hungarian company didn't maintain the patent validity, and they could return home very happy. But it was not the end of the research. It was a lead compound, practically the lead compound, which you see on the right side, timotrazole, was the sulfoxide derivative of it. And it was very important to develop an in vivo test, the drug fistula model, which was very useful in the drug discovery. The problem was the serious side effect was observed, the enlargement of the thyroid gland. The analog optimization, nevertheless, could eliminate these side effects, to make a long story short. And here you can see the omeprazole, the successful product, which has a characteristic substituent, methoxyl on the para position of the pyridine, and in the position 5, another methoxyl in the benzimidazole ring. And nearly parallelly, in Bikulden, or parallelly, Bikulden and Hesselwatt people discovered the mechanism of action of this very interesting compound, which is a prodrug, which prodrug on the effect of proton gives a sulfonamide, a very unstable metabolite. And this is the active metabolite. And this story, which is very interesting, because high throughput screening is very good and useful, but on the other side, it cannot be used in such a drug discovery, because if you see, if you have a great amount of compounds and you find omeprazole-like agent, it is a prodrug which is inactive in vitro. On the other hand, this metabolite cannot be found because it's very unstable. And that is the reason that in this case, let's say, this type of methodology doesn't work, wouldn't work. Let's have a look on this figure. I received this from my Otsuka colleague from Japan. It was prepared by the Japan Health Science Foundation. How they see the present year of 2015. And here the horizontal line shows the therapeutic satisfaction and the vertical line the therapeutic efficacy. And if you see, the drug research has great achievements. The peptic ulcer, nothing is perfectly solved, but it is nearly solved. problem. It's a great achievement. The same can be said more or less true in the case of the antihypertensive agents, where we have at least 7 drug classes are used. Then you can see a lot of other things which are even not completely solved, but more or less use the treatment possibility to have again a good quality of life for the sick people. As gout, angina pectoris, hyperlipidemia, asthma. On the other hand of the figure, in the lowest corner, you can see Alzheimer, age-related macular degeneration, lung cancer, and so on. It is sure that we have a lot of tasks yet. Based on this figure, I think we will have to do a lot. But on the other hand, we have to recognize that we have already a lot of achievements behind us. To make a short summary, I wanted to give some highlights from drug research of the 50 years, which was nearly impossible to make in a lecture, but I tried really to show you that target-based drug discovery can be very successful. And I mentioned 5 examples where the lead compounds and the drug compounds differ in interaction. It is very serendipitous. Thank you for your attention.